In 1953, Wilkins and Franklin got very fine X-ray diffraction photographs of DNA, and they showed that the helix has a width of 2 nanometer. So the distance between two strands is basically 2 nanometer, and one turn of the helix is 3.4 nanometer. With 10 layers, or you can say 10 step type of thing in a helical stepways. So 10 pairs of bases are stacked in it. So it's 10 pairs of bases stacked in it. James, Watson and Crick worked out the first correct double helical model of DNA, and they were awarded Nobel Prize in 1962 for building the 3D model of DNA called as the B model of DNA. So there are more types of models. There is A model also, C, D model also, Z model also. We'll be talking about in brief regarding them also. So now this brings us to the next topic, and that is the Chargaff's rule. So what exactly is Chargaff's rule? Chargaff in 1950, he made observation on the bases and other components of DNA, and these observations are today called as the Chargaff base equivalence rule. So base equivalence means the number of purine will be equal to number of pyrimidine. We all know purines means A plus G. So in a given piece of even in a given double-stranded DNA, purines and pyrimidines are always equal in their number. That means A plus G is equal to T plus C. This is purine and this is pyrimidine. And this means what the molar amount of A, because we all know that A will be facing T and G will be facing C. This is all you have done in the 11th class already. So the molar amount of A will always be equal to the molar amount of T, and the molar amount of G will be always equal to the molar amount of C. Then next point, the sugar deoxyribose and phosphate occur in equimolar proportion, because we all know the backbone of the DNA, or whether we talk about RNA even, the backbone of the nucleic acid or a polynucleotide chain is made up of alternating sugar and phosphorus. So that's why the sugar and the phosphate occur in the equimolar proportion, and the ratio of A plus T to the G plus C is constant for every species. We need to remember this point also, that A plus T divided by G plus C will never be equal to 1. Please remember that. It is going to be variable for different organism, because though we say A plus G is equal to T plus C, this is correct because purine is equal to pyrimidine number, but A plus T number could be high in some organism, could be low in some other organism. So this ratio is constant for every species, and this helps us to identify the source of the DNA. So this is used a lot in evolutionary study, even in the forensic science, whenever there are animals which attack the people. So there are many other things also in which this ratio help us to identify the source of DNA. If a crime scene is there, and if some sample of blood or DNA is obtained from there, then this ratio does help a lot to understand the situation which occurred and what exactly has happened in the crime scene. So let's understand the next point now, that the deoxyribonucleotide is the DNA. It's a heteropolymer, and it's a polydeoxyribonucleotides, and it is made up of deoxyribose sugar, nitrogen bases and the phosphoric acid. Nitrogen bases are of two types, purine and pyrimidine. Purines are adenine and guanine, and pyrimidines are cytosine and thymine. Adenine always faces, or you can say makes the double hydrogen bond with thymine, and guanine makes 3 hydrogen bonds with the cytosine. This is the rule for double-stranded piece of DNA according to Chargaff. Watson and Crick model of DNA always spoke about the 2 strands of DNA, and these 2 strands of DNA, they run in a right-handed helical manner. And remember, according to their model, the DNA has 2 strands which run anti-parallel to each other. So today you will understand diagrammatically that why do you say one end is 5' end and the other end is 3' end, because the same digits were used even in the case of the RNA. So they are anti-parallel, that means they are running, if one has the head on the top, the other has a tail on the top. So 5 prime to 3 prime, and the other strand runs in 3 prime to 5 prime direction. Each strand has alternating sugar and phosphate. That's why we learned the number of the sugar and phosphate would be equal. And alternating sugar and phosphates are there making the backbone. And nitrogen bases are stacked inside the helix in a perpendicular manner to form base pairs, because whenever there is one base here, the other strand will have a base pair just opposite to it, following this rule: A will face T and G will face the C with double hydrogen bonds, or otherwise triple hydrogen bonds. So number of purine will be equal to pyrimidine, or you can say when one strand has purine, the other strand will have opposite to it pyrimidine, or otherwise vice versa. Then A will always face T, or T will face A, and G will always face C, or C will always face G, and the number of the hydrogen bonds is fixed. According to their model, one turn is equal to 3.4 nanometer, and one turn has 10 base pairs. So the distance between two successive base pairs would be 0.34 nanometer. The distance between the two strands would be 2 nanometer. Each nucleotide turns 36 degrees from the previous nucleotide in horizontal plane. So it turns in this manner, in a right-handed helical manner, and it will be each nucleotide, if one nucleotide pair is like this, then the other would be at 36 degree, so slightly turning in a helical manner to make complete 360 degree turn with the help of 10 base pairs. The Chargaff rule in 1950 was given by Chargaff for double-strand DNA, and according to him, number of A is equal to number of T and number of G is equal to number of C. That means purines, because these two are purines and these two are pyrimidines, will be always equal to the number of pyrimidine. So A plus G divided by T plus C is equal to 1. But remember, this ratio is not going to be same for everybody. A plus T divided by G plus C is the ratio is low for primitive organism and high for the advanced organisms.